name is Jesse Coe. As Amber mentioned, I'm the business development representative. I cover Western United States, Western Canada, and South America. But I did just recently move to this position. So prior to that, a little over 4 years at KBI in our analytical department. We call it Analytical Formulation Sciences, or AFS. And before that, I think that was that plus some of the time I had before in grad school, postdoc, a couple other stints in industry. Marked about 11 years in thinking about and dealing with proteins and macromolecules. And so one thing that I think is kind of undeniable about them to me is they're very individual, they're very complex. They could be stochastic and difficult to work with, but also really interesting, really powerful. So kind of that's the segue into what I'm going to talk about today, which is product characterization and control strategy. A little bit in a general sense, but really what I'm going to focus on too, and the latter half of this talk is on a case study, in particular for an antibody drug conjugate, or an ADC. KBI provides comprehensive characterization services ranging from early stage through late stage development, and we've got platform approaches to make sure that we're timely and successful for filings and things like that for our partners. And this includes for ADCs as well as other biological moieties. This talk is based upon the understanding that an effective control strategy is based on regulatory expectations. It's also a good business practice, quality and efficacy in therapeutics, and then it's developed through comprehensive understanding of the product attributes themselves. So we have really, really good expertise across KBI and some of our cross-functional teams to help our partners deal with this from early stage of preclinical all the way through marketing applications. So without further ado, a quick overview of KBI. For those of you who may not be familiar, a few sites across the world. We are a JSR life sciences company, global CDMO. Our headquarters is in North Carolina. This is actually at Hamlin Road, where I'm at right now, is our first site, but we've, as you can see, grown to a couple more sites in North Carolina, 2 in Colorado, and then 1 in Geneva, Switzerland. We're focused mostly on liquid drug substances in both mammalian and microbial space. We do partner with Argonaut for our fill finish and liquid drug products production. We kind of do an end-to-end offering. We currently manufacture the carrier proteins for ADCs, and we have years of experience in non-cytotoxic conjugation. We're not currently able to support cytotoxic conjugation in-house, but we are actually building the capability to do so. So stay tuned on that. I also want to mention our collaboration with our sister company, Crown Biosciences. They also cover some of their things that are a little earlier than that. So early discovery, translational services, and this also includes ADCs. We also have extensive analytical capabilities. So my shameless plug here, we've got a new portal that's up and running, and it's great. So anybody who's looking to do any kind of straightforward analytical work with us, this is somewhere where you can go in, kind of request a quote. We'll be able to get you a quote back. We can sign it through there, take care of sample submission forms, and you can track progress of your testing and even get the results on there. So it's kind of one spot to get everything. And this usually happens within a few weeks from the initial request. So it's a really, really nice way to get a quick turnaround on testing. So as a CDMO, we support our partners in a wide variety of ways. It's kind of evidenced here. I'm not going to go over every single one, but we cover, like I said, cell line to market. And then within that portfolio, in particular, what I'm going to talk about today is our site in Louisville, who, among other things, specializes in product characterization. So from a regulatory standpoint, product characterization is a necessary part of enabling both INDs and BLAs. Beyond regulatory requirements, this is a crucial process to really understand your products. This informs all kinds of things. It gives us justification for setting specifications. It drives process strategy, and it ensures efficacy and patient safety in the end. So today, I'm going to focus on this for an ADC, and I'll start with a little bit of background before I kind of jump to that. Some of you may already be really familiar with this, but for those who aren't, I want to kind of define what product characterization is and why we care. Why is it necessary? And hopefully by the end of this talk, I'll have answered that question for you. So it's defined in regulatory guidance such as ICH Q6B. It involves comprehensive analysis to understand physical, chemical, and biological properties of the macromolecule, or the therapeutic product, I should say. It's not just macromolecules that would need this. And for our purposes is the characterization of heterogeneous forms and the impact of that on product quality, and in addition to that, kind of pathways of degradation are of interest as well. KBI, each characterization activity is associated with one or more detailed technical reports. For a comprehensive package, we would then consolidate these to elucidate to provide an elucidation of structure summaries. This is a section that would go in your filing, as well as an assessment of attribute criticality. This is something that's going to help you set and justify specifications for your process. I do want to note the bottom line, it's distinct from process characterization. So they sound similar. They're certainly quite different. Product, we're talking about the drug substance or the drug itself versus the process to make that. And so these are both very important large endeavors, but I think sometimes it's not as clear if haven't been around a lot. So I'll kind of go down the list here somewhat quickly. But you can see on the left-hand side, it's kind of some of the stages or activities that are really involved at a high level for characterization. Top one is primary structure, and kind of the main point of this is to ensure the fidelity of the DNA's transcription. Sequence variants can be present, and if they are, this is something that can certainly impact product quality and safety, and so it's something that needs to be understood well. This can also create regulatory concerns. The next couple points are talking about primary structure and so other variants, glycoforms, charge variants, etc. So these also need to be characterized to understand if and how they're impacting product quality. For late stage, you need to know this for any impurities that are up to or that are above 0.1%. So the threshold's pretty low. You really need to understand all the things that are going on. Early stages, I think they usually set this closer to 1%. As you move forward, understand your process and your product better, you really need to understand all of those impurities and whether they are impurities. Moving along, secondary, tertiary structure. We've heard a bit about this today, but this is also very important to ensure proper folding, among other things. So misfolded proteins very likely to impact product efficacy, safety. Critical for us to monitor that. Biological characterization. So this is kind of to do with method of action, potency, these kind of things. So pharmacokinetics, cellular signaling, and any immunomodulatory effects that could be present. So these are, of course, quite important as well for both safety and efficacy. And then the last one down here is degradation pathways. And so this is important in a lot of ways. And you've probably heard of things like a forced degradation study, and that's exactly what this kind of falls under. These studies will look at any relevant stress conditions. This is typically things like photostress or acid and base, oxidation, reduction. As John was mentioning earlier, if you're got a syringe product, then you want to think about tungsten or silicone, anything that can cause any issues or any impact on your product quality. Thermal cycling, obviously, another one that's quite common there. This is also needed to really understand and use to gauge if your methods are fit for purpose while you're identifying critical attributes. And so what I mean by that is you need to understand if any points in the process you've identified critical attributes. You need to understand if you can actually see these things. You can see the aggregates, the deamidation, fragmentation, anything that may be critical needs to be quantifiable and well measured by your methods. And so this is a way to generate those things and tell if you're going to see them, if they're things that are going to arise or increase on stability or be important to the process. So this is the case for that's the case for all biologics, but for ADCs, there's an extra layer of complexity. This is due to some of the intermediates and kind of a non-trivial modality. I'll talk about the structure of that in a little bit. I think you'll see why if that's not already popping in your head. So to kind of relate product characterization to your control strategy, I touched on that a little bit, but there's four main areas where this inform kind of happens. And they're listed up on screen here. So this product characterization is something that will be completed throughout the drug development lifetime. I'll show that kind of against the timeline and where those activities kind of happen. This starts with screening studies. You need to understand developability. You need to characterize reference standards. And then again, even the early IND needs some characterization for elucidation structure and other properties. Comparability, stability assessments, degradation pathways, these are all kind of part of that. And having that understanding enables you to make a quality target product profile. That's QTPP. number 2, and that's going to help you kind of really understand where those critical attributes are. This is, of course, you can't state it enough, required for regulatory filings. This is not optional, right? So you need to do this. You really should be scientifically as well, but you have to. This is a requirement. And then to fill that out, part of that, and I alluded to it a bit, is being able to justify and set your specifications and understand, what variables need to be controlled when, and can you actually monitor those. One of the keys to success is avoiding a certain element, and you can't find it in this table. Does anybody have a guess what that might be? It's surprise. So you don't want any surprises, right? Especially as you go later into the stage, you need to really understand what's critical and how you're looking at it. And so, I've got a few things up here: sequence variants, variable results, etc. But these need to not be surprises. When you get towards validations or your late stage characterization, these need to be activities where you are effectively confirming what you already know. This is not the time for you to test and experiment and learn something new. That really leads into this point. You need to start this process early. Characterization is not cheap. It's not a cheap thing to do, but it's a lot cheaper than waiting to the last minute, doing it poorly, and having those surprises and finding things out. And it's expensive, not in dollar amounts, but in time, in effort. So having to correct these mistakes later on, rather than understanding them and avoiding them in the first place, is going to save you in all kinds of areas, right? Establishing these methods, analytical test methods. Anybody who's done GMP work in a late stage has probably seen one or two methods at least, where you're going, how did this happen? How did this get this far? And how is this issue here? We're missing something, and there's a surprise, and you see the pain that is. Deviations, change controls. These things are expensive for time and money, and making sure that you're able to hit your filing deadlines. And all companies, if you're looking at runway, there's all kinds of things that are domino effects of that. So I hope I've convinced you this is something that it should be invested in early and done right, and make sure that you're doing it with a team that kind of understands and what needs to be done, has that experience to do it. So I said I would look at this versus the timeline. This is an accelerated timeline. It's actually pretty typical for ADCs at this current climate are on these. But you can imagine, the analog of this looks quite similar for a traditional timeline. So you need to start early preclinical. Again, you need to support some of this with the IND with some of its knowledge. So platform approaches are usually good there. You do want to make sure that you're coming at with as much knowledge as possible. So throwing a good wide net without being too wide. And what I mean by that is, looking at the literature, looking at similar molecules, any experience that you have, you need to understand, what things do need to be looked at, and either that you can check them off early as they're not an issue, but also explore to make sure that you're starting that process early. As this kind of moves forward and you start going to clinic, you need to really be optimizing these platform methods towards your molecule, because again, these are complex, they're stochastic, right? They're hard. So those methods need to be kind of evolved to really enable you to zone in on your specific product. And then as that happens, you kind of have this iterative process where you're informing yourself. With that, you're able to kind of keep optimizing these. But prior to getting to late stage, you need to prove it, right? You need to make sure that these methods are in their final state and they're, do a fit for purpose qualification. This is not your QC method, so you're not going to have a validation of these, but this is kind of analogous to that. You need to be showing that these methods are doing what they need to do. They're covering everything. You don't have any outstanding questions that you could be asked, or that that you don't know the answer to, that could affect something in your product when it's going to patients. And then late stage, of course, you do need that section for your BLA filing, this characterization testing, and this is where you need to be confirming what you already know, right? At this point, you need to absolutely understand where things are at. and what's critical and not, and just be showing that. For the rest of the talk, I'll kind of apply that. Now we have a bit of background on product characterization. Let's look at an example, and in this case study is an ADC. This characterization was performed at Louisville and led by the team there. This, we had some background on dealing with ADCs and doing some characterization, but this was the first comprehensive ADC project for KBI. And much of this work, this ADC specific, was done in collaboration with CMC scientists at ADC Therapeutics. So I do want to also thank and acknowledge them for allowing us to share their data, their success story. Obviously they were an integral part of this, not just throwing money at us, right? They were a partner. This case study is going to kind of be structured with these bullet points on the left here. We'll go ahead and kind of take a high-level look at the molecular structure of this. We'll discuss what kind of an ADC is from that point of view. We'll also kind of talk briefly about the process overview, how that's made, because that's actually going to have some effect on what we need to do and think about with understanding what attributes and what the things we need to look at are for it. I'll touch on the ICH guidance. So small versus large molecule, because an ADC, I think John called it a Franken molecule, but that's kind of an apt description, right? So this has a small molecule moiety and a large molecule moiety. So how do we deal with that, right? There's stuff in place in the industry for both these at this point, but this is kind of a little bit new in that regard. And then I'll talk about attribute evaluation. You'll keep hearing me say that, but also, kind of within the framework of how experience can help you guide that plan and make sure that you're successful at it. Then I'll talk about considerations and establishment of these methods. And then we'll get to kind of the meat and potatoes of this, which is the elucidation of structure, and I'll show some of the data, and the critical quality attributes assessment that we're able to make from that data that's kind of informed by that. So I do also want to mention before I get into this, when I'm showing data, is representative of the actual data that we collected, but of course this has got some proprietary information. So certain things have been masked. You'll see trends and things like that, but this has been altered slightly to make sure we kept that proprietary information confidential. So on to molecular structure. So some of you may not have seen this or thought about this before, but what is an ADC? And I briefly talked about it last slide, but for this particular one, this was a humanized IgG antibody. The isotype was specific for a cancer cell antigen, and the effector functions were engineered out for this one. This was then connected to a polymer extension group and a linker, and then the cytotoxic drug or payload. If you hear me say payload, that's what I'm referring to. And this is the kind of small molecule moiety going on to the antibody. The antibody has typical components that you would expect for an IgG. It's got CDR region. There's the inter and intramolecular disulfide bonds. There's glycosylation. And of course, as macromolecules that I alluded to, right, they're expected to have multiple variant forms. Part of this is due to variable post-translational modifications. And this creates a mixture of slightly different molecules or microheterogeneity. To further compound that, so for an ADC, the way that this particular one, and I think it's common across ADCs, was conjugated is through a thiol reduction in the hinge region. I don't know if it's always the hinge region, to attach this, right? I think there's some click chemistry involved in there. And the intermolecular disulfide bonds have a maleimide conjugation, the polymer extension, and payload after that. So this particular image, you can see the 3 red dots, right? That's our payload. And so this example has 3 conjugated sites. But this number can vary between 0 and 8. So there's an extra layer of stochasticness to this. And so one thing that I'll talk about in this as well, if you hear me say DAR, that's the drug-to-antibody ratio. So this would be a DAR of 3, right? There's 3 drugs to one antibody. So to talk about the ICH guidelines, again, so we've got this large molecule, we've got the small molecule. These are governed by different ICH documents, right? 6QA is a little bit older, right? Small molecules have been around a lot longer. And then as biologics kind of came to prominence, they've added guidance for this as 6QB. And these have different considerations. So smaller molecules, in a word, are better behaved, right? They're a lot more homogeneous, pure. They need less thought into, well, I guess they need a simpler solution to keep them stable. Their buffer or their matrix doesn't necessarily need to consider as many factors. And a lot of the methods for looking at them are quite well established and pretty easy to look at, different molecules with the same approach. Biologics are complicated. I'm probably going to say that a few more times. So when you think of this all together for a little bit, I think it becomes clear that, what really makes sense here is to move to the biologics. So this Q6B is the relevant framework to look at this because you really do have that kind of complexity, that heterogeneity, and that doesn't really mesh well with this small molecule. So really what your strategy becomes is to look at this from the biological perspective. So here's kind of a quick level view of the process for making this. And so, of course, you're making this in a couple of stages, right? So instead of making just a drug substance off the bat, you need to make a couple of intermediates before you conjugate those. So you have a drug substance intermediate that is your MAB, and then you have a drug substance intermediate that is your small molecular linker moiety. And you need points of control on both of those, and then those come together and you conjugate the intermediates, and that's when you've got your final drug substance. And then for this product as well, the drug substance differs from the drug product, and that's in that it is a lyophilized product. So drug substance then gets lyophilized. There's a couple of points of control that that really need to be looked at, and that is characterizing and understanding the large molecule, the monoclonal antibody on its own, the intermediate. And then we want to look at the drug substance. So after this conjugation's happened, we want to understand if there's any differences, if we've had any effects from that conjugation process. We also want to consider that lyophilization step. So I think I'm missing a star there, but that also needs to be looked at because that can affect things. So understanding those 3 points, that's really where your points of control need to be, and what needs to be considered when you're doing a strategy for characterization. Like moving back to the structure and thinking about this, in a general sense, regulatory guidance, prior knowledge from literature, experience with similar molecules, these all help define the approach to characterize an ADC. This approach can be similar between products using the same MAB and same linker drug conjugate, but generalizing that too much further has some pitfalls in it. So care must be taken. Even a single amino acid substitution has the potential to impact activity and stability, sometimes significantly, just from the MAB. And different spacers, linkers, payloads, even the number, the DAR changing can drastically impact product quality. And I'll actually show you that in a little bit. So again, it's complicated and some thought must be taken, but generalizing is something that you should do only when you're really sure it's appropriate, and make sure trust but verify even on that, right? So here's a few other considerations within that. This is further compounded by multiple mechanisms of action. The antibody is responsible for going and finding the target, looking for the antigen in the cell in the body to destroy without harming your healthy cells. And then the drug is responsible for killing the cell once it gets there. There's also potential for an effector function. I'm not going to cover that here because this one, as I mentioned earlier, this map had that engineered out, but that's extra complexity as well that's possible for ADCs. So characterization is important to understand what attributes are critical for preserving these mechanisms of action. Some critical attributes are well known for this type of ADC. The maleimide conjugation is pretty well studied. It's susceptible to a deconjugation by the retromichael mechanism. and can lead to off-target toxicity. So we want to make sure, we're not cleaving this inappropriately or before this payload is near its target. And so ADCs also, you can imagine, you've got a small molecule next to a big one. mAb is, of course, pretty soluble, but small molecules oftentimes are pretty hydrophobic. And so this means that ADCs in general are known to be prone to aggregation. And this can lead to loss of potency, can have the potential for these aggregates to become mutagenic. And so this is another thing that kind of, in general, you really want to assess and pay attention to with your characterization strategy. We'll kind of go through some of the biochemical attributes and methods and some of the ones in this case. So as they have more heterogeneity than mAbs alone, there are more attributes to consider with regard to criticality and assigning product-related substances versus product-related impurities. So understand the difference between these. Again, published data and knowledge of the product helps to create a list, methods to detect them, and then a justification to include that attribute in a control strategy. But we need to listen to the data. So here we see highlighted post-translational modifications in the CDR region of the mAb, this N-terminal glutamate. Actually, screen sorry. This one shows, the PTM and the CDR, which can be critical due to potential interference with binding the target antigen. And this can increase to critical levels with storage and handling, as would be something we would see in stability studies. We can determine which methods would be important to characterize the PTMs and also which QC methods we can use to detect this. For this case, peptide mapping, CIAF, of course, these are some things that would help us kind of understand this and be able to monitor that. Disulfides and free thiols. Another well-known critical attribute is the disulfide structure, and obviously this is key to the conjugation process itself as well. So we need to understand how that's impacted by the conjugation, how the molecule looks before and after that, and does this, at the end of the day, of course, we're always asked, does this affect our product quality, and is this something that is affecting stability? So, it's structural integrity there. Incorrect pairings, disrupted bonds, these can lead to misfolding, aggregation, degradation, structural variants may lead to unwanted immune response and pharmacokinetics, as I've kind of said earlier. Free drug, right? The toxic payload and related impurities are also well-known critical quality attributes. We want to make sure that this cytotoxic payload is where we want and is released when we want. So understanding that and monitoring that for safety and the linker stability with any kind of relevant stressors you would have in your process until, it's where it needs to be and get released. Aggregates. Aggregates are well-known CQAs. I think I've already said that a few times, but this is something that needs to be monitored for safety, efficacy, immunogenicity can, easily come from this, or loss of potency. It's important to have orthogonal methods to test for aggregates. So to ensure that we're seeing them all, again, this is making sure that we're able to see that 0.1% and really kind of separate it and understand it. So the QC methods themselves need to be able to do that. In this case, AUC was used to look at this aggregation. It had a better suited range for isolating, identifying the aggregates. It was able to resolve them more clearly than the more standard SEC approach. Amide hydrolysis is a well-known thing. This is well understood. It's described in literature, and this can impact stability. This hydrolysis can kind of come from the enhanced stability. So actually, hydrolysis can lead to enhanced stability of the maleimide thiol linkage, which is going to be kind of surprising. Glycan distribution consistency is a regulatory requirement. So this is obviously, as it is that because it is well known to be a critical attribute that needs to be monitored. So efficacy, safety, these are kind of the main drivers for this, right? And pharmacokinetics as well for that. There are other attributes to include in this, so the DAR and some of the other ones that I've mentioned, but I think this gives a pretty good high-level idea of what you want to look at for these. Some method complications, especially for this, right? So some specific considerations for an ADC need to be taken into account just due to the nature of the structure there. So with a conjugated DS, there's potential for it to nature. We need to think about that when we're doing methods like CSDS or reverse phase, where the environments, these during the sample prep or the analysis itself, may by nature show different profiles than the mAb alone, just because of how those environments affect the molecule. So the probe is effectively impacting the sample itself. Similarly, for intact mass, non-denaturing conditions must be, verified and used. Other things on that you would see. So any kind of method that you have, you'd understand the sample stability. Effectively, you may not have the same method you can use for the mAb intermediate that you can use for the ADC. You have new considerations, and you need to make sure that these things are not affecting that, or if they are, that your method is optimized or even, changed to a different technique to make sure you're accommodating that to see what you need to see without, affecting the results just by the process that you're doing. So pH and heat sensitivity are a couple things. Light, that payload could absorb light within UV range, and that's something that's the basis for a lot of methods that you see and think about with your maps. So that needs to be taken into account, and you need to understand where that absorbs and how that interacts. And then hydrophobicity, I already mentioned, denaturing, propensity to denature, these kind of things, aqueous colloidal stability. So the same matrix may not work for your mAb as it would for the conjugated antibodies. There's a lot of things to kind of think about and consider when doing this. Test methods are developed for both. If something works for both of them, great, but that's certainly not the expectation or the rule. So you can see here an example of a reverse phase LC method. And we've got the DS in the top, where we've got a little more interesting of a profile there, whereas at the bottom we've got the mAb intermediate. And this kind of looks like a standard mAb for anybody who's looked at these type of things. But we can see for the ADC, we've got a lot of different species up there. And so now we need to understand all of these and characterize them. So non-covalent interactions can be disrupted during the separation due to the naturing properties of the acidified organic phase. So the same methods, you're not going to have comparable profiles. It's kind of the end of the day idea. And so you need to look at both of these separately and understand what those impurities might be. So looking at the elucidation of structure, so this is kind of a quick blurb from the section from the regulatory, and kind of what I want to draw your attention to here. I want to kind of point out, so impurities, I've talked about this a bit, and then justification, specification. These are two other sections that can be informed by this work. So this characterization towards the elucidation of structure is not decoupled from these, right? By doing these activities and kind of understanding what your impurities are, necessarily, that's going to, of course, inform your impurities section. And with this knowledge, you're going to start to understand what's critical and what's not, what's changing, what are stability indicating, and that's going to help you to form that specification and justify it. So this is a pretty good chunk of this CMC documentation for filing. One thing that's notable, and I guess was maybe a surprise in some ways to the team, was that you need your own section for the mAb and for the conjugated DS. It's an extra regulatory complexity that's added for this for ADCs. So they're approachable, but they're a challenge, right? There's a lot of things that you think about a little bit differently. Certainly, as this is still kind of a current and emerging therapeutic, strategy that's still, being fleshed out. It's the path's not as well trod. Primary structure, characterization, expected amino acid sequence was confirmed. Let's see, show that. And the N-terminal pyroglutamate was there as expected. There's no C-terminal lysine residues, heterogeneity. related to antibody and the conjugate. PTM levels, they're comparable between the DS, DP, and the MAB intermediate. So that's great to see. We're not seeing loss or anything else that needs to be kind of investigated, and the process is not altering this in kind of an irrecoverable way. The conjugated DS demonstrates the expected heterogeneity. I've given reasons for why we expect that, right? For native cysteine conjugated ADC. So here's kind of a few different DARs, right? So you can see little red dots. I know it's kind of small, but you can see some different ratios. And now you can see just the expected heterogeneity, of course, comes from this. This is something that we expect from the process. And so you can see this hydrophobic interaction column chromatograph on the bottom right here for the conjugated ADC. Signals at 250 to accommodate this. And the conjugate is used to determine the number of conjugates under each peaks. This is a way to find out what that DAR distribution is in your product. Moving next to glycan characterization. Glycan analysis generally controlled at the MAB level. Conjugation is not expected to change the glycan distribution. Of course, this needs to be verified, and it was. So there wasn't a difference in that. And that allows you to, you don't need to look at glycan at every step, every time. You've been able to assure yourself that this isn't impacting it. You do need to look at that, but you're able to look at that at the MAB intermediate level now. So that goes back to cost and time, right? Like you want to make sure you have the right tests, but you don't want to add tests to what's needed to release and move things forward. Glycan analysis showed the presence of one low-level, potentially immunogenic species, which means that we can see that. We know what it looks like. It's there at a low enough level that this is not an issue, but you know that that's something critical to watch, right? Kind of move on to the next one, the CX chromatogram. So that's what I'm showing here for the antibody intermediate. And so it's pretty cool. You can see the profile for the DS up there on the top left. You can see 7 peaks right there numbered. So in order to characterize these, and this is something that's done with any of these methods, this is kind of the example, you would understand, how many peaks do you need to isolate them? And then you need to kind of check those always against potency at least, but anything else that you think may be critical. So in the bottom left, you can see those re-inject on that same method just to prove that you've got the species you need there, that you intended to. So the fractions are re-injected there and confirmed to be those peaks. And then on the right, you can see kind of the results on there from the potency. And so we can see a couple of these forms are low potency forms. So these would not be drug variants. These are impurities, right? These are something that we need to. These are critical attributes that we need to understand and monitor. The good news is, they're minor, and 2, most of the forms showed nominal potency for that. These 2, it looks like one of them lost complete potency, and this is due to a PTM and CDR region. This makes sense, right? Moving on to biophysical characterization, the near UV CD up in the top left. This is only showing this for the MAB, and the reason for that is that the payload itself is actually interfering in that region. But we do see kind of the expected tertiary structure for the MAB. Again, unable to test them with DS or DP due to interference in that region. Far UV on the top right shows that the secondary structure is highly comparable between the intermediate and the DS. So it's determined no impact from that conjugation. And then on the bottom, we've got the DSC thermogram. This shows the overlay between the 2 formulations for the intermediate and the DS. And we do see a few differences here that I kind of want to point out. So the first one is at T onset. There's a small destabilizing effect from the conjugation. I can actually see. Yeah, so there's yes, I should clarify. There's 2 formulations for the MAB intermediate. That's the gray and blue. I'm not sure they're super well resolved here, but they're also quite overlain. And then the red one is the conjugated DS. We can see that kind of differs in a few spots. That onset's a little bit earlier with a little less thermal stability. This is typical, expected. It's not an alarming shift, but we see that start to lift off a little bit earlier there. There is a slight shift in that TM1 that first transition point, the large peak, as far as where that happens. This is related to the Fab region, but it really is pretty comparable. The main thing that you kind of notice is for both the gray and blue, you see that shoulder off the back side of that main peak. And that's, we're calling that TM2. Obviously, the red line doesn't have one there. So you call that TM2 for that, but that is going to be related to the CH2 region. And that's where the conjugation occurs. So this is not expected. This makes complete sense, but we definitely do see a difference there. And then the TM3s are highly comparable again, kind of realigning it there. So we understand, from literature and experience, what that TM2 is. So it's not alarming that we see that difference. It's kind of expected there. But again, this is where it kind of comes into play with using all the tools at your disposal to really understand and digest these data. All right, so stability indicating methods from the degradation pathways is really cool. So this is aggregation again. It's known to be a likely CQA. This is true even in regular biologics, but certainly for ADCs for some of the reasons I've mentioned. So doing an orthogonal check to make sure on this, compared the AUC and the SEC data. So one of the goals is to make sure that the QC method, which is going to be SEC, is detecting the aggregates that form during stress conditions. We can't actually use the SEC method for our QC. The results from each method are compared against each other. You can see AUC is on the x-axis and SEC on the y-axis for some of the various stress conditions. So these data points are from different types of stress. But what we're looking for is a linear correlation here, and that's exactly what we see. So we're able to convince ourselves that this is a suitable method for us to look at this. Stability method, indicating methods from degradation pathways. And so all the QC methods, I showed one example of that, but were found to be stability indicating. So they had been developed the proper way and were able to capture the important points for this and justified. The example here shows that despite the relative stability of the MAB intermediate, the SEC detected these changes in the accelerated thermal stress condition. This is at, I think, 40C. And we can see this over, 5-week period where we're able to see the dimer, these low molecular weight species kind of form and grow. So this is kind of the other way to look at that data. So at the end of the day, and obviously there's a lot of work I'm not talking about here, but this really is a really large effort. This allows you to kind of make a table and understand, okay, we've marked our CQAs, what bins are they? Which ways do they need to be monitored? Do we have a method to do it? We've shown that. Is this, where points of control do we need to do this at? As I mentioned earlier, some things we were able to look only at the MAB intermediate. Some things, we need to look at multiple points or at all points possible. And then, how to justify that and allow you to really set what that risk tolerance is for those. So being conservative, certainly you want to minimize risk. This is important for patient safety and efficacy at the end of the day, but only including the necessary points of control. So by doing this, we're able to reduce, rather than like a full scale, or I think of the right word, instead of doing absolutely a full permutation of what could be done, you're able to pare down and do the things that are the minimal set, which is still as effective as possible and as needs to be. The CQAs were determined using this risk assessment. This is kind of a small snapshot, so I do want to mention that this is obviously not a full table, and this is an example here and a bit of a high level, but it gives you an idea of kind of what that looks like at the end of the day. So with that, again, I want to acknowledge that I'm presenting this work. This was not something that I did. So this is really the big team here that was involved in this. I know there's others that are probably not mentioned on here that also contribute. This kind of work takes a huge multifunctional team. And I do want to, I think I missed the main point of that is this was successful. This has gone to market. So we were able to enable that BLA, the filing. This has become, a market product at this point, which is one of the first ADCs to do that. So this was a Herculean effort. It's over an 18-month time point. Yeah, so I want to highlight the team there and just show it really takes a lot to do this. It's a complicated project, but the expertise is pretty impressive. So with that, thank you guys all for listening.